they make themselves stable in the host environment, okay, and how they replicate. So all these, you can say, processes, they are collectively we termed as a bacterial pathogenesis. In the context of the virus, the same things applied. Okay, how virus enters in our body? It will enter through different parts, which we already discussed, starting from the mouth, okay, tiered skin, and there are other respiratory pathway, okay, the urinary genital pathways. These are the some of the parts, okay, and how these you can say the virus will reside in us, okay, they replicate and they disturb our normal physiology. So that's the meaning of viral pathogenesis, okay. And just remember that viral will replicate, viruses only replicate, okay, in host body, okay. Viruses only replicate inside the host body, or you can say in the host body, okay. They are, when you suppose this is the duster and here are some viruses, okay, they cannot multiply on this, you can say, duster, even though you provide the nutrition, but they won't you can say replicate. But in context of bacteria, if you provide their specific requirement, that's the nutrition, they can, you can say, replicate. But in context of the viruses, they won't replicate, okay. They need the living host body, they need the living host body. That's why the virus culture is impossible, okay. You cannot harvest this virus outside, okay. So viruses culture can't be performed, in vitro, means outside the body, okay, outside the body. So viruses can replicate only inside the living cell. Now, if we talked about the virus pathogenesis manifestations, their manifestations lies mainly in the cellular level. So consequently, the viral pathogenesis manifestation can seen at cellular level, okay. In the cellular level, either they can, you can say, inhibit the host, you can say, genetic material, or either they make cell lysis or cell death, or either they change the normal, you can say, host cell into the malignant cell. That's termed as the transformation, which we are going to see, okay. But the things which we have to remember is that viruses only replicate in the, or we can say, the inside the living cell, okay. And consequently, the first manifestation of viral infection are seen at the cellular level, okay, seen at the cellular level. Now, these manifestations, okay, initially they are acute, and termed as the acute viral infection, okay. Initially, these manifestations are acute, okay, and these acute infections can be, you can say, discarded, or you can say overcome totally by our immune system. But if our immune system are, you can say, compromised, okay, they are not strong enough to fight with these virus infections, then there will be a persistent infection. There will be a persistent, or you can say, prolonged infection. Got the point? So initial exposure of virus infections lead to the acute, you can say virus infection condition, and that infection is more or less overcome by the immune system. But sometimes what happens, these acute viral infection, they follows the persistent infection. Okay, they follows the persistent infection. Understood? No problem. Now we understood the mechanism, okay, that the viruses are, you can say, only replicate in the inside the host body. It means what? Why only they are only replicate inside the host body? It means we have to discuss about the virus, their morphology, their physiology, you can say. So let's make the heading, write the viruses. Viruses generally devoid, okay, cellular structure. They won't contain too much complexion. Okay, you can say the complexity of the body organization is absent. Okay, they won't carry, you can say, the too much cellular organelles. They don't have the too much cellular organelles. That's why they can replicate fast. They can replicate the fast. They won't have, you can say, too much cellular organelles. If we talked about the viral, you can say, organelles, they contain mainly, sometimes they contain the cellular envelope. They have their infectious particles, that's called as the genome, their genetic material, that's the infectious part. And for protecting those genome, the viruses have the capsid. So mainly the genome, capsids, these two things are present, okay, as a cell, you can say, cellular structure in the viruses. But sometimes some viruses have external, you can say, envelope. Okay, so those viruses are called as the enveloped virus, and the viruses where these envelopes are absent, they are called as the naked virus, or also called as the non-enveloped viruses. Okay, non-enveloped viruses. Now, so viruses are what? They are the small, okay, or if we talk about their genetic material, first thing, they are small, okay, far more smaller than the bacteria. Okay, now the another thing is, if we talked about their genetic material, so either this bacteria have the RNA, okay, or either they have the DNA. Both they don't carry RNA. You know, it either they have the single stranded RNA or double stranded RNA. RNA won't be double stranded. It cannot be the double stranded. Okay. Now the DNA, it maybe have the single stranded DNA, or it maybe have the double stranded DNA. If we talk specifically to the humans, if we talk specifically about the humans, then the most common form of virus, you can say, genome, which affect the humans, they are either single stranded RNA or double stranded DNA. So this single stranded RNA and double stranded DNA type of virus are more, you can say, most causative agent in the humans. Clear? No problem. Single stranded RNA and double stranded DNA. It's about their genetic material, and viruses need, remember, it need the host to replicate, to, you can say, divide. Now, just discuss the structure of the virus. As I said, that virus devoid, okay, they won't, you can say, contain so many complexity, and that's a one reason to replicate at a faster rate. Now, you can say the viruses, they have their genetic material, okay, they have their genetic material, which is, you can say, protected by the envelope, which is protected by the envelope. Okay, just draw it with different colors so it it will look good. It's their genetic material, also referred as a genome. Okay, and these you can say the round structure as you are seeing, this is referred as a, yeah, this is referred as a capsid, and this individual you can say one round structure, they are a protein, you can say coat, they are protein coat, and one individual you can say protein coat, they are known as a capsomer. So you should write it down that this one unit is called as the capsomer. Okay, and this entire ring of you can say capsomer, they are called as the capsid. They are called as the capsid. Fine. And this genetic material, it is referred, yeah, it is referred as a genome of viruses, viral genome. No problem. Yeah. Now this you can say, as I said, that the some viruses have you can say neck structure. You see, this is only the capsomer, yeah, and the genome is present. Okay, genome is responsible for causing a infection. Okay, and this capsomer is meant for protecting these genomes. Okay, no problem. Now, but in some of the viruses, these structure, they are, you can say, enveloped. They are enveloped. Okay, they are, you can say, covered. Fine. And these viruses, they are called as the enveloped viruses. They are called as the enveloped viruses. And in this enveloped membrane, there are, you can say, enveloped proteins are present. Enveloped proteins are present. And these enveloped proteins, they are meant for protecting the virus as a whole. Okay, no problem. Now, along with the capsid and genome, they are called as, they both are the infectious part of a virus. So they are also called as the virions. Virions. So how you define the virions? Virions is a infectious part of virus. It's a infectious part of viruses. Got the point? No problem. Now, and this type of viruses are called as the enveloped viruses. Okay, this type of viruses are called as the enveloped viruses. No problem. Yeah. Now let's summarize it once again and then we will move ahead. What we discussed about viral pathogenesis, and we said how viruses causes a disease in human by means of, you can say, increasing their number, increasing their progeny. Okay, and that we term as a viral pathogenesis. Fine. Then we said virus need a host to replicate. They won't, you can say, replicate in non-living objects. Fine. They won't replicate in the host living, you can say, non-living object. They need a host to multiply, to propagate their life. Okay, the another thing we understood, okay, since it's required the host body, it means we cannot culture it in artificial medias, okay. Then we seen that consequently, if it need the host to replicate, it means it will cause signs and symptoms, as we can say, their manifestation at a cellular level in a different way. Either they can make cell to die, or either they transform the cell, fine, or either they make fusion. Okay, that's referred as a cell fusion. Then we said that the first incident of this viruses pathogenesis is like a acute. Okay, it's a sudden. Okay, and that is most of the time encountered by our immune system, but sometimes immune system failed. Okay, that result into the persistent infection. So generally what happened, the acute viral infections follows the persistent infection. Then we discussed about the viruses, that viruses are, yeah, non-living entity which become living in the host environment. And it is also a borderline of, you can say, classification between the living and non-living, because they won't carry those, you can say physiological system. They won't have too much complexity in their, you can say, body organization. So that's why they can replicate at a faster rate. Now they are small, and if we talk about their infectious part, okay, you can say the most infectious part is their genetic material, that is referred as the genome. Genome is a whole set of their genetic material. So their genetic material can be RNA, okay, or it can be the DNA, but both are not present in the same time in a one virus, okay. The RNA is a single stranded RNA, or DNA may be the single stranded type or double stranded type. Then we make one conclusion that the single stranded RNA as well as the double stranded DNA, they are the most causative, you can say disease causative type of viral genome in humans. Then we discussed the structure of the virus. The virus structure is very simple. They have their genetic material that we referred as a genome, okay. Then they have one protein coating that is referred as a capsid, and the function of this capsid is to you can say make to protect the genome, fine. Then we seen that in some of the viruses there are one external envelope, okay, and these two things are, you can say, present in all the viruses. But this envelope, it can be present or it cannot be present. So it may be present or it may not be present, okay. And this envelope is referred as a envelope membrane, and this envelope membrane is decorated by the envelope proteins. Fine, till here no problem, understood? Now let's move ahead and discuss about that how this, you can say, a virus, okay, how this virus will replicate, which way, like in which pattern they replicate, fine. And that is, you can say, dealt in the viral replication, we seen the bacterial replication, we can say we seen the bacterial divisions, how bacteria